So let's establish the overall purpose of the Calvin cycle. In the simplest terms, the Calvin cycle is building a high energy organic molecule or molecules. Remember, organic means that it's a carbon-based molecule, meaning that it's made like its skeleton basis is carbon, and then you can add other atoms onto that, other elements. So carbon-based, for example, something like glucose is a classic example of something people say, oh, photosynthesis makes glucose. That's the carbon organic molecule, the carbon-based molecule. And this is through two kind of steps in the Calvin cycle. So one thing that we're going to do is to attach carbon dioxides, CO2s, to make the carbon skeleton. That's going to make that organic part and the actual molecule. But then to make it high energy, we do the second part. We're going to embed electrons. Little e with a minus sign is a symbol for electron, which is negatively charged. So embed electrons in the bonds of the molecule, and that's what gives you high energy. So it's all that stored energy in the bonds. Okay, let's draw the basic outline of the Calvin cycle process. We have a starter molecule. This one happens to have 5 carbons in it. I'm just going to call it the starter molecule. And this is what we end up with at the end. This is why it's a cycle. You end up with the same molecule. And we're going to take that starter and attach a carbon to it in the form of carbon dioxide. So there's a single carbon for our CO2. And we'll stick that on the starter. Right there. Made a bond. So that's step number 1. So attach CO2 to the starter molecule. And we call that attachment fixation. So we're doing carbon fixation. We stick the carbon on through the as carbon dioxide. All right. Then this molecule happens to break into 2. So it's still all the carbons there though. Our next step is we said we also need to get the high energy. We don't want to just build carbon skeletons, we need to get high energy. So in order to do that, the first thing is that we need to make these molecules here reactive by adding a phosphate. I'm going to draw that in orange because that's my ATP color. And that's where the phosphate comes from ATP. There's the P, 3 phosphates on an ATP. So let's show that happening. There's our ATP. And where did this come from? The light reactions. There's a video about the light reactions. If you haven't watched that, you can. So T is for tri, meaning 3. D is for di, meaning 2. Why is there 3 and then 2? Because we used one of them. And what did we do? We stuck it on the end to make this nice and reactive. I'm just going to draw one of these, but the same thing happens to the other one. So now we have this highly reactive molecule, and why did we do that? Because we wanted to get electrons in it, so we've got to make it reactive first. How's this going to work? Well, we're going to embed electrons in the bonds. And the general strategy is you're going to switch out the phosphate that you just stuck on there 4 electrons. And the electrons are from NADPH that we made in the light reactions. Okay, so let's draw that. So we have NADPH with the electrons coming in, dropping off those electrons, and it leaves as NADP+. That's the cab that's empty, meaning that the electrons are not in there anymore because they came out and came into the molecule. So we've still got those 3 carbons, and then the electrons are, sitting in the bonds here. It's just a representation. And we said what we had to do in order to get the electrons in, we had to switch it out by popping the phosphate off. So the phosphate pops off and those electrons can jump on in. So now what we have is a 3-carbon compound with electrons embedded in it, and that's our goal. It's a high energy carbon compound or organic molecule. And that organic molecule can be converted to glucose first, and then that can be converted to other carbohydrates, which can be used to help make parts of proteins and lipids and nucleic acids and all that stuff. Now the thing is, we said that this is a cycle, so we have to get back to the beginning. So when this is going on, there's actually a lot of molecules doing this process at the same time. So only some of them leave the cycle to go be converted to glucose and then go, be used by the cell. The rest of them will get converted back into this starter molecule. So that's this 3rd step here. So you're regenerating the starter molecule. And then you can start it again. And this is the very basic version of the Calvin cycle part of photosynthesis.